I'd like to welcome everybody to SC Embry's first Thirsty Thursday. This is your chance to quench your thirst for knowledge, learn a little bit about what's going on across the Embry network research-wise, and hopefully have a chance to chat with a few people, maybe folks you know, maybe you don't. But the idea is just to kind of bring us together periodically. Incentive is that John Clarkson is going to try to come up with a mocktail for every Thirsty Thursday. So that way we're going to stretch his culinary skills. And so for this one, Dr. Rawlings, who's our speaker tonight, his favorite drink is a mint julep. So John was challenged with coming up with a mocktail version of a mint julep. So hopefully you all, or some of you at least, took advantage and tried to make your own mint julep. As I was traveling today, missed out, but I will participate next time. And so I'd like to introduce our speaker for tonight, Dr. Jason Rawlings. He was a DRP faculty member in Embry 3, correct? And has since graduated, has done wonderful things. And so we wanted to invite him back. He's at Furman University, and we wanted to invite him back as our first speaker to kind of catch us up on what's been going on. It's been a while since we've heard from Dr. Rawlings. So with that, I will turn this over to him. I guess since it's really, I'll see if I can, why don't we hold questions to the end, kind of unless you prefer to have people interrupt along the way, or how do you want to?
It doesn't matter to me. Either way works. Whatever works for everyone is fine with me.
Okay, well then it's a small group right now. So I guess if you have questions as we go, just kind of, you should be able to unmute. If you can't, I'll have the chat open and can fix that. But if you have a question, then as we go, we'll just keep this casual and you can speak up and ask. So with that, take it away, Dr. Rawlings.
Well, thank you so much for having me. I really appreciate being here and having the opportunity to share the work of my students that have been working with me for, going on a little over 10 years now at Furman. So my research lab is interested in understanding how chromatin changes its architecture during an immune response and how that change can impact immune cells' ability to proliferate during an immune response. So what you're looking at here on the left, this is a naive T lymphocyte. And so this is a cell that has not yet encountered its pathogen. It's very small, it's very quiescent, it's G0 in the cell cycle. But when it encounters its cognate pathogen, it undergoes this dramatic change. And so obviously the cytoplasm gets huge, but what we're actually interested in my lab is the changes in the architecture of the nucleus. In particular, we go from this very condensed chromatin to a very decondensed chromatin. What I'm going to hopefully show you today is that this actually has huge impacts on the cell's ability to express genes, particularly those required for cell proliferation. So I'm going to talk a little bit about T cell proliferation, in particular, how two signaling pathways are involved in regulating whether or not a T cell will proliferate during an immune response. I'll then kind of give you a one-slide summary of my postdoctoral work at St. Jude Children's Research Hospital, where I showed that, in fact, it is the status of chromatin that will determine whether or not a lymphocyte can respond to growth factors. And then I'll spend the bulk of my time talking about kind of two general thrusts of my research lab at Furman. First is looking at the signaling pathways that control the decondensation of chromatin, and a more recent project of mine looking at how chromatin status might regulate other lymphocyte populations. I'll end by talking about some of the more future directions of the lab, and I also want to introduce another project that I've never actually presented at Embry before. So give you a little teaser of something else that's on the burners, as it were. So as you know, the immune system is our defense against foreign pathogens. And the way it works is that it has the ability to tell self from non-self. And things that are non-self are going to get attacked by the immune system. And of course, the immune system has a lot of different weapons at its disposal. Today, we're going to talk about one of those weapons, and those are T lymphocytes or T cells. Now, there are multiple subsets of T cells. We'll talk about some of those a little bit later. But during an immune response, they all kind of have really three phenomena that will happen with regard to these cells. They'll all become, T cells can become activated once they encounter their cognate pathogen. That causes them to differentiate into what we call effector cells and begin proliferating, okay? And so I'm gonna focus on those two in red, the activation and proliferation. Now, clinically, we see evidence of this proliferation when you get sick. So if you're, for example, if you have strep throat, you go to the doctor and they'll fill you up just under your neck. They're looking for your lymph nodes and see if they're swollen. That swelling that they're looking for is in fact that lymphocyte proliferation. And so you might be asking how we study this, this phenomenon. So it turns out Furman administration doesn't take too kindly for me to do experiments on undergrads. Apparently has issues with, causes issues with enrollment. So instead I use the laboratory mouse as my model system because their immune system is very similar to our own. So as I told you, there are two pathways that control lymphocyte proliferation. We're going to talk about both of them. The pathway on the left is what controls the activation aspect of T lymphocytes. And then the pathway on the right is the proliferation response. And so this begins when an antigen is presented to the T cell receptor on the surface of a T cell, which will activate a number of signaling pathways, which we'll get back to in just a moment, leading to the expression of the IL-2 gene. This will make the cytokine IL-2, which can be secreted from that cell. It can bind to the IL-2 receptor, activating the JAK-STAT cascade. STAT5 is the primary transcription factor that gets activated in this process in T cells. It can translocate to the nucleus, where it can drive expression of genes responsible for advancing the cell cycle. So we have the pathway on the left that's involved in activation and the pathway on the right for proliferation. So now putting this into context, so in each and every one of your lymph nodes, you've got millions upon millions upon millions of T cells. Each one of these T cells has a specially engineered T cell receptor that allows it to recognize a very specific pathogen. So if this schematic were to represent those millions of T cells, you might have some that can recognize influenza, some that recognize Streptococcus, E. coli, or even the virus that causes COVID-19. Now, let's say you get sick or you get exposed to COVID-19. We want that particular T cell that is equipped to recognize that virus to become activated. And of course, as I just said, it will produce interleukin-2, leading to the proliferation of that cell. These cells will produce more interleukin-2, creating more cells, and so on and so forth. It's kind of a chain reaction, as it were. And so this is what causes your lymph node to swell. Now, the question I had regarding this is that in this environment in the lymph node, we have all this interleukin-2, you have all the cell proliferation. The only cells that are proliferating are those that are specific to SARS-CoV-2 in this case. And so I wanted to know, why is it that these naive cells, the ones shown in green, how are they able to ignore the IL-2? Now, of course, since we're talking about control of proliferation, understanding these mechanisms can have some very far-reaching consequences. Namely, if we don't control proliferation appropriately, we can get some very nasty pathologies like cancer, immunodeficiency, as well as autoimmune disease. And so here's my one slide to summarize about 5 years of my postdoc. What I was able to show is that in naive T cells, they possess all of the machinery to transduce the IL-2 signal. So in other words, we will get activation of STAT5. It can translocate to the nucleus. However, it does not have the ability to bind to DNA. It doesn't actually engage the DNA. And the reason for this is that the DNA and the chromatin that package it are in a very condensed configuration, as you see here. Now, in the context of an immune response, when antigen is presented to the T cell receptor, we'll activate those signaling pathways, causing that change in chromatin. This allows STAT5 to access promoters of target genes, and you get expression of those genes responsible for advancing the cell cycle. And so immediately was interested in trying to understand the mechanism behind that. And so for this, I first want to return to that first image that I showed you on the first slide. And this is that naive T cell, and this is the activated T cell. And of course, you can see the differences in the chromatin condensation status. But what you can't appreciate from this image is how do you get from here to there? And so we did a time course analysis of this, looking at various time points post-activation. And as you can see, the cell does undergo some very dramatic changes, but the changes in chromatin happen relatively early. Even with the naked eye, you can begin to see changes at, say, the 3 to 6 hour time point. I was able to develop an assay to look at this biochemically by looking at the solubility of histones. And so in naive cells, histones are not very soluble, and so we can't detect them with the standard Western. But as a function of activation, they become more soluble, and we can detect them. And of course, this has direct impacts on the cell's ability to express genes. And so we're looking at one STAT5 target gene. And so in naive cells, IL-2 has barely any ability to induce its expression. But after 1 hour, it goes up. After 3 hours, even more. And that little star means that it's statistically significant. And by 6 hours, even more. And this last one is the equivalent of this last cell over here. And so So what we can see here is as the cell is decondensing its chromatin, it is gaining the ability to express those genes. So one of the things I did was develop a flow cytometry assay to test and allow us to rapidly analyze chromatin condensation status. And the way the assay works is that we take antibodies that recognize those histone proteins and fluorescently label them. We put them onto cells, and if the chromatin is very condensed, very few antibodies will bind to their epitopes, giving us a very weak fluorescent signal. If the chromatin is decondensed, more of these antibodies will bind, giving us a much larger signal. And so we can actually measure this using a flow cytometer. And so we actually wrote this up as a protocol. This is Megan Lee, a student of mine, former student of mine, and Kelly Bingham. That's actually Megan Hanna now. She's married. She got married a few years ago. And that's what I look like when I used to have hair. So when I'm just going to take a step back here and say, when I give these talks like this, and I realize I should probably not use pictures of myself, because I realize how old I'm getting now. A lot less gray and a lot more hair there. But the good news is those pants still fit. All right, so back to it. So the flow cytometry assay is really cool because it allows us to analyze tens of thousands, even hundreds of thousands, even millions of cells at a time. And not only that, it gives us a quantitative measure of our histone accessibility, or the condensation state of chromatin. And not only that, it's sensitive enough that we can even detect changes as little as 15 minutes post-activation. So this decondensation of chromatin is a very rapid event. So we set out when I got to Furman to try to figure out the signaling mechanisms that control the decondensation of chromatin. And so when a T cell receptor gets engaged as presented antigen, two main signaling pathways will get activated through the hydrolysis of PIP2 by PLC gamma 1. We'll get the production of IP3, which signals to increase intracellular calcium, ultimately activating NFAT. Whereas the other product of that hydrolysis is DAG, which can activate PKC, which will then activate downstream pathways NF-kappa B, as well as the MAP kinase cascade. And so one of the first experiments that Megan and Kelly did when they joined my lab was to activate each of these pathways individually and ask whether or not either one of them could induce the decondensation of chromatin. So we can use the reagent PMA, which activates PKC specifically, or ionomycin, which causes an increase in intracellular calcium. And what they were able to show is that either one of these reagents is able to induce the decondensation of chromatin, which means both of these pathways seem to be involved. They were joined by Jenna Meredith, now Sermons, and Taylor Mitchell, now Jones, to further characterize the role of calcium in this process. And we published this back in 2015, and they were able to show that calcium, specifically from the endoplasmic reticulum, intracellular stores, is both required and sufficient to induce chromatin decondensation in alpha beta T cells. But what's very interesting is that although calcium can induce the decondensation, it does not make cells competent to respond to IL-2. In fact, that's the job, it seems to be the job of PMA, shown here on the right. So that's the PKC side of the pathway, which suggested to us that maybe these pathways are actually working together in this process. And so to test this out directly, we realized by reading the literature that there are a number of isoforms of PKC that are in fact calcium dependent. So we decided to test the hypothesis that maybe it was a calcium-dependent PKC that was causing the decondensation of chromatin and the subsequent competence to respond to IL-2. The way we tested this was to chelate intracellular calcium with BAPTA-AM and then stimulate the cells with PMA to activate PKC directly. And what we were able to find, and this is Molly Shields and Sarah Eichenbusch's work, is that when we chelated intracellular calcium, we reduced the cell's ability to decondense chromatin. This was validated by Jen Funston and Kenny Murillo Brizuela, who looked at gene expression. And of course, when we chelate intracellular calcium, the cells are unable to express that target gene. So these data suggest that a calcium-dependent PKC is what is required for decondensation. So we then turned our attention downstream of PKC at these two pathways, NF-kappa B and MAP kinase. So I'm not going to present the NF-kappa B because that's negative data. And so instead, we're going to focus on MAP kinase. And so we're going to use a very similar approach as before. We're going to use an inhibitor of MAP kinase signaling. It's called UO126, not to be confused with the Van Halen album OU812. Rest in peace, Eddie Van Halen. And so we inhibited MAP kinase with OU126 and then stimulated the cells with PMA to activate PKC and asked whether or not we could see gene expression. So this is work by Jen Funston, Tia Scott, Haley Swatzel and Audrey Ward. And as you can see, when we inhibit MAP kinase signaling, our ability, PMA's ability, or PKC's ability, excuse me, ability to make cells competent to respond to IL-2 is diminished. So I'd like to switch gears a little bit and talk about the kind of the second main thrust of my lab, and that is to ask the question of whether or not the mechanism that controls the decondensation in alpha beta T cells is also utilized by other lymphocyte populations. And so a number of lymphocytes all arise from the same developmental precursor. These include natural killer cells, other subsets of T cells, which we'll get to in just a moment, as well as B cells. And our hypothesis is that perhaps chromatin condensation may regulate these cell types as well, because not only are they derived from the same developmental precursor, they utilize some of the same signaling pathways for their activation. So this is work that was spearheaded initially by Sierra McDonald, who is now a PhD student at the University of South Carolina in Columbia. Also work done by Elena Knobb. This is Jamila Johnson, Chelsea Joseph, and Morgan Fretwell. And they were able to show in B cells also, just like T cells, possess a very condensed chromatin, but upon activation, it becomes decondensed. They were also able to show that when we chelate intracellular calcium, we can delay or at least mute a little bit that decondensation event, suggesting that perhaps calcium is working in the same way in these cell types. Jeremy Ersick and Ryan Scruggs, as well as Sarah Stewart and Haley Sabota. Haley actually joined us from the Citadel for a summer and worked on this project. They were looking at NK cells shown here on the left, as well as gamma deltas on the right. So we'll talk about the NKs first. NK cells also possess a very condensed chromatin in the naive state. As a function of their activation, they also decondense their chromatin. And as you can see, they do so to a much lower extent than alpha beta T cells, which is actually reflective of their proliferative capacity during an immune response. So these cells don't proliferate as robustly as T cells do during an immune response. Over here, we're looking at two subsets of gamma delta T cells, those that are expressed at the developmental marker 1.2. So these are known as naive gamma delta T cells. They look very similar to alpha betas, both in the basal state as well as when we activate them. Those that express lower levels of Thi 1.2 are considered to be memory or previously activated gamma delta T cells. They have a higher or a less condensed basal state of chromatin that becomes even more decondensed as a function of activation. And this turns out that these patterns that we see here are also indicative of what we see with regard to their proliferative capacity during an immune response. So these data are very intriguing because they not only show us that these cells also change their chromatin as a function of activation, but gives us some indication that they might be using chromatin as a way to regulate the kinetics of that proliferative response. Back in 2019, that was the last summer I had on campus before COVID shut us down. I had the pleasure of working with Cassidy Ron and Madison Hurst, who continued to work on both gamma deltas and B cells to ask whether or not the PKC and the IP3 pathways could decondense chromatin in those cell types. And just like alpha beta T cells, both our gamma deltas and our B cells can be activated by selective stimulation of either the IP3 pathway, the calcium pathway, or the DAG pathway, giving us a little bit more of a premise to, or a little bit more evidence to suggest that maybe they're utilizing the same signaling mechanisms as well. So I've covered quite a bit. We've talked about chromatin status and how it regulates alpha beta T cell proliferation in response to growth factors. I showed you some evidence that suggests that both DAG and IP3 signaling are very important. In particular, it appears to be a calcium-dependent protein kinase C that acts through MAP kinase to make cells competent to respond to IL-2. And I've given you some data to suggest that other lymphocyte cell types, namely B cells, gamma delta T cells, and NK cells, may also utilize chromatin to regulate their proliferation as well during an immune response. So I'd like to just end by talking a little bit about kind of where we're headed in the future. The first one is we want to see if what we saw with alpha beta T cells is also true with those other cell types. Namely, is it a calcium dependent PKC that regulates decondensation and competence to respond to growth factors? The second main thrust I have is I've been very curious about what happens during development. So today we've talked about what happens during activation, this decondensation of chromatin leading to this burst in proliferation. Well, it turns out T cells, which arise in the bone marrow, actually travel to the thymus where they mature. And when they enter the thymus, they have a very high proliferative capacity. Their chromatin is also very decondensed. But as a function of their maturation in the thymus, their chromatin becomes more condensed and they lose their proliferative capacity. So I'm very interested to see what the mechanism is that's behind this condensation event. Now at Furman, unfortunately, I've not been able to do these experiments because our flow cytometer that we've had for the last 10 years simply was not capable of doing those types of analyses. But I've been very fortunate that I was just recently awarded a National Science Foundation grant to purchase a brand new flow cytometer, which actually arrived on Furman's campus yesterday. So it's still in a crate. So I'm waiting for them to unbox it and get this thing operational so I can get this work done. So I'm really excited about that. I'd like to end by throwing you just a little vignette of another project that I've been working on at Furman as a side project. It's a collaboration with another member of the faculty at Furman. His name is Brian Guess in the chemistry department. He is a former INBRE target faculty. I think he was in INBRE 2, if I'm not mistaken. Anyway, this project has to do with another signaling pathway that gets activated when T cells encounter an antigen. And so when that antigen binds to that T cell receptor, I mentioned a lot of pathways get turned on. One of them is the PI3 kinase pathway. What PI3 kinase does, it acts on that same molecule PIP2, but what it does is it phosphorylates it, forming PIP3. PIP3 can go on to activate another molecule called AKT. AKT is a very important molecule in the cell. It regulates cellular metabolism, growth, as well as survival. And it turns out that many cancers will hijack this pathway by hyperactivating this pathway that gives them that metabolic advantage that they need to continue to grow. For this reason, back in the day, before HeLa cells and whatnot were developed, scientists actually used to use activated T cells as a cancer model. And so you can imagine then, because of its roles, PI3 kinase signaling, its roles in cell metabolism would be a great target for chemotherapeutics. It turns out that there's a really, really potent inhibitor of PI3 kinase called workmannin. I've got the structure down here at the lower left. Workmannin will bind to the ATP binding pocket of PI3 kinase, forming a covalent bond and inhibiting its enzymatic activity. Now, workmannin would be a great chemotherapeutic drug. Unfortunately, it's very unstable in neutral buffers. It falls apart very easily. And it's also promiscuous, so it binds to other proteins in the cell, which would give it some pretty nasty side effects if this were to be put into humans. Brian's lab at Furman, they've synthesized a much simpler version of workmannin. This molecule is called hibiscone C. It has this electronegative surface here. This is a furanosteroid. Has this electronegative surface. This is the part that actually binds to that ATP binding pocket. Now, this molecule is more stable, a lot more stable than workmannin. And so we decided to see whether or not it had biological activity. So this is a paper we published back in 2017. This is work that was done by Caroline Besley, Dina Reinhardt, Taylor Emmons, and this is Brian Guess over here. And so as you can see from this Western, in our cells that are T cells that are growing in culture, they show an activation of AKT. If we give them the vehicle that we dilute our drugs in, they still have activated AKT. But if we inhibit them with workmannin or hibiscone C, we block the activation of AKT. So our current work on this project is to make modifications to hibiscone C to not only understand how it binds to the PI3 kinase ATP binding pocket, but also to make a more potent version of this molecule, and also one that is very less promiscuous. So I'd like to end by acknowledging all the great work and by all the great people that have worked in my lab. I think I've acknowledged many of them along the way. This is a photo of my lab last summer. We worked all virtual. That's Ilias Wahibe up here. This is Chase Hudson. This is Roy Vanderplow and Hagen Nelson. We actually just submitted a paper based on their work. It's currently under review. I also want to thank South Carolina INBRE because everything that you saw here today wouldn't be possible without them. As you can see, all of these publications have been possible because of support through various INBRE mechanisms, including the DRP. Also want to thank the National Science Foundation for not only the MRI that's going to allow me to do work going forward, but they've also provided me some funding along the way. Finally, I also like to thank NASA. I just got a grant from them this past year to study the effects that microgravity might have on chromatin status in lymphocytes. So this is through the South Carolina Space Grant Consortium. So it's kind of like the NASA version of INBRE. And finally, I'd also like to thank Furman University. And so with that, I'd like to stop and take any questions that you might have. Thanks again for having me.
All right. Well, thank you, Jason. I appreciate it. You had me laughing at several points in time you could not see. But,
I like to put some humor in there. You know, I like to keep it light. This was casual.
Yes, and this is supposed to be sort of a, you know, an informal-ish time for us to get together. And so everybody can unmute their mics so we can have some discussion or questions. I'm going to start with one.
Sure.
So I was wondering, I don't work in the immune system. I look at fibroblasts.
Okay.
But fibroblasts undergo what we would call an activate. They become activated.
Mhm.
They go to a myofibroblast phenotype, and it also changes their proliferative capacity.
Mm-hmm.
And so I was just wondering, have people, or do you know if folks have looked at the idea of this chromatin condensation, chromatin remodeling as a way to regulate activation in other cell types?
You know, I'm not aware, looking at in terms of an activation phenomenon, I'm not aware of that happening. The thing with lymphocytes that's so amazing is that they are I don't know if this is true for fibroblasts, but when you take them out of the animal, they're in G0, and they've been in G0 for decades. And they won't budge until you stimulate them.
Mhm.
I'm not sure if that's true. I've played a little bit with fibroblasts. Like I've made MEFs before back in the day, mouse embryonic fibroblasts. That's about all when I was back at St. Jude, and that was, 15, 16 years ago. So I'm not aware of that. I am interested in looking at other cell types. So while I was at St. Jude, there was a guy there was working on red blood cell development. And so they, before red blood cells, the precursor cells eject their nuclei. The nucleus does become condensed, and we were able to get a little bit of preliminary data on that. It wasn't overly convincing, but it does appear that it is happening. I think the big issue with some of these cell types is that trying to get an assay that allows us to see those differences. So like in red blood cells, the reason why I say it was very iffy, if you look under a microscope or at the nuclei, like if you just do like a DAPI stain, you can easily see those TER119 precursors really clench up. But if you do a Western for like histones, you don't see a change. And so, and I think the reason for it is, how they're condensing and what they're doing is a bit different than in T cells. And so trying to figure out those conditions, like for that Western blot, to get the conditions right to where you can actually see that difference is a bit difficult. So, one of the things I can do, for example, is if I were to put lysis buffer on those cells overnight, even naive cells will eventually succumb to the lysis. So it's all about finding that sweet spot. And as long as you can find it, you can do these types of assays. But I'm not aware of any other cell types where this has been done this robust. It's a great question, though. I would be very interested if other people are seeing these things.
Jason, can you tell me a little bit more? Can everyone hear me?
Yeah, I can hear you.
Okay.
Yeah.
Can you tell me a little bit more about that Western that you did? I'm really interested because we used to do some histone work where we were looking at hormone modifications of histones. And I know that we had to do a really, really harsh lysis, and we would just get all the histones out.
Yeah, so let me do something. This is really cool. So you can't do this, because you don't necessarily bring stuff with you to a conference. But let me bring up my EMBO paper where that Western actually came from, because I only showed you part of it there, and I want to show you the rest of it because it's going to come right back to what you're talking about there, because we had the same problem, and I'm trying to click on folders and talk at the same time. This is not going well for me. So I can't walk and chew bubble gum at the same time either. All right, so I've got it. Let me bring up this is a paper I published that kind of launched this whole thing back in 2011. And so I'm going to show you, where is it? Right here. Yeah, so when we were doing our Westerns, right, initially, so we were actually our initial hypothesis was maybe chromatin modification, like histone modifications were going to be the key to this whole change. And I started doing these Westerns where I was looking at various modifications like H3K4. Are you familiar with the terminology up here? Monomethylation of lysine 4 H3, right? And I was seeing this, no modification here, a whole slew of modification there. And of course, I initially thought, oh, wow, yeah, it's histone modification. But every single modification I looked at, it showed the exact same thing. Even if these were modifications that were associated with gene activity or modifications that were associated with silencing. And so it's when I did the total histone Western, you can see I saw the exact same thing. And that's what clued me in that it wasn't a matter of, histones being modified. It was a technical issue, right? Our ability to detect those histones. So then I came back and did an acid extraction. And that's like the harshest thing you can probably do to histones because they're, being basic proteins, you can really get them out. It's just hydrochloric acid and they come out in spades. And so, yeah, I mean, the conditions are really tough. If, using this is an NP4, I don't know what kind of buffer you're using. This is an NP40 based buffer. So I don't know if you're using RIPA or other buffers, but
I think back that when we were doing like the histone extraction, so we could look at modifications, I think it was a urea based
a urea, okay.
one we had gotten from the literature that
Yeah.
we did the Western initially. We couldn't see the histones. So
yeah. Well, because you were running into the same thing. So what kind of cells were you looking at?
We do ovarian granulosa cells. They're the ones that are around the oocyte, and they change a lot. And of course, they are hormonally driven, and they have a increase in hormone FSH and LH. They cause a increase in several different types of modifications. Of course, acetylation being one of the most prevalent. But those cells do go from a more condensed situation in their chromatin, their nucleus, to a very large euchromatic nucleus, prominent nucleoli, because they become little steroid factories.
Okay.
And I would think that, there's some similarity to the cells you're looking at, even though I know it's a completely different system,
Right.
because, with those lymphocytes, you've got all that machinery being hyped up to make T cell receptors, or if you're looking at B cells, I guess this happens in B cells too.
Yeah. So if you're making antibodies, absolutely.
Yeah. All that machinery to do whatever that cell is supposed to do. Plus, they terminally differentiate once they hypertrophy.
Right.
But you can see them with a very condensed nucleus before ovulation, then after ovulation, just a huge nucleus. The whole cell triples in size.
Oh, wow. Yeah, that is really cool. I would not be surprised at all if you're seeing the same things that I'm seeing here. Now, is that something you can trigger in vitro? Do you culture these cells and you can stimulate them with hormones to induce these changes, or do you have to extract?
Yes. So to really turn them from the early cell to a later cell, it takes about a week and adding a lot of hormones. You can accelerate it if you put the cells in serum, and that has growth factors that kind of move things along faster.
But it takes about a week or so to see that change.
To see the dramatic change from, and one of the things we measure is our endpoint is progesterone. So, after a day, you're getting tons of progesterone, but if you look at these cells after a week, you're getting like 1,000 times more.
Oh, wow. Yeah, no, that's very interesting. So, for these, for T lymphocytes, as you saw, it's about 24, 48 hours gets you pretty decondensed. And it takes them about 24 hours before they enter the cell cycle as well. So, I mean, they're really having to crank up just to be able to, to do, really basic things. That's very interesting. Now, the cool thing about our flow cytometry assay, we can detect very subtle changes. So if you would be interested in seeing how quickly that happens, if it's a rapid change or not, we could do that by flow. Absolutely.
Oh, cool. I'll have to look more into that.
Yeah, I'd be happy to help you out with that for sure. That would be a really interesting collaboration. I've never worked with those types of cells before, so that would be cool.
Hey, can you guys hear me?
Walden. Yeah, I have a maybe two related questions. Can you hear me okay?
Yeah, I can hear you.
Oh yeah, wonderful. So really good mechanism for T cell activation upon chromatin decondensation. So I'm just wondering, in the naive state or in disease state, how those two PKC or a calcium signaling pathway are somehow regulated differently. so that T cells will be activated differently?
Yeah, that is the million-dollar question, Walter.
Okay.
I'll be honest, because we are seeing differences in these cell types, and so the question is, are they utilizing these pathways differently, and if so, how is that possible? How is that happening? One of the things that we can't do at Furman that I would be very interested to see is, are they flexing calcium to the same extent? And it could be something as simple as that. Now, one thing that I haven't presented today is that, I told you the NF-kappa B data was a bit negative. That's not entirely true. There is a very subtle effect with NF-kappa B. We've also looked at NFAT, and it's a very subtle effect. And so it could be that there's cumulative effects amongst multiple pathways. But you're absolutely right. Trying to figure that out for sure.
Okay, thank you.
I have a question. Can you guys hear me?
Sure can, Jason.
Hey, Jason, I'm Jason. I'm sorry you can see. I cannot for the life of me figure out how to work this damn camera. But I loved your talk. I think we used great, and I have a question specifically regarding your B cell data.
Yes.
It gets me thinking about one particular question that always pops into my head, and as I'm exploring lymph nodes and high affinity antibody responses, and one of the big questions in the field is, what's the major difference in short-lived versus long-lived plasma cells? What are the fundamental features that B cells responding to the same antigen might decide to shuttle down the short-lived plasma cell? Rapid proliferation, immediate production of IG. I love your talk. I think it might decide to enter a germinal center and go through somatic hypermutation and affinity maturation, all these things. And do we know anything about what the condensation of chromatin looks like and how it differs between SLPCs and long-lived plasma cells?
No, we
Does anyone
don't
know anything about that?
That's, as far as I know, that's not known. And so my old flow cytometer, the one I had, was an old, if you're familiar, it was a fax sort. Came off the line in 1993. Only has 4 colors on it, so I couldn't even do those assays if I wanted to. I didn't have enough colors. But our new machine, we can. It's a 3 laser system, 12 colors.
Oh wow!
Yeah, it can do 35,000 cells per second. So yeah, we're gonna be. That thing's gonna be on fire. It's gonna be awesome. But so no, I'm absolutely, and I'm also interested in looking at what's going on in the bone marrow with hematopoietic stem cells, all of that. I'm really interested in all of those things, and I haven't even thought about looking at those things because I didn't have the machine to do it, but now I do. I'm really, I'm like a kid in a candy store now. But yeah, I totally agree with you. I think it'd be very interesting to see if those subsets are regulated differently.
Can you selectively target some kind of promoter or inhibitor of chromatin decondensation to specific cells? Is there a system to do that?
Well, no, I'm not sure. I mean, the only way that we can control decondensation, the way we've been able to do it is just pharmacologically by looking at these various inhibitors of the pathway. So the most potent thing that we can do right now is use intracellular calcium chelators like BAPTA-AM, which seems to be very potent. No matter what we do, when we throw that on cells, that really causes the chromatin to pucker up. Now, how we would direct that to individual cells, I have no idea.
So they've been doing this. So there's a guy named Gabriel Victora, and he's like, I got a professional crush on this guy. I've had it for a decade now. The guy does amazing work in B cell biology. He really comes at it from an evolutionary perspective. I love what he does. But he's doing things to target germinal center B cells in order to manipulate high affinity antibody production, and he's using antibodies targeting DEC-205. So I was thinking maybe there's a way to target some kind of inhibitor or promoter to start manipulating chromatin condensation status in GCB cells and see whether or not you could differentially influence high affinity antibody production. So there's ways to target those cells specifically if you've got the inhibitors that could be conjugated to that anti-DEC-205 antibody.
Yeah, I'll definitely take a look at that. That is awesome. Yeah, that would be amazing.
Yeah,
we definitely need help with that. Our animal facility isn't really big, so to be able to do that.
Absolutely. No, yeah. So in terms of collaboration, yeah, anytime you want to think about getting into B cells, and let me know, because I'm definitely interested in the ideas in how condensation regulates what's going on in the during selection.
Absolutely, yeah. So I'm very naive when it comes to B cells, pun intended. I'm a B cell guy, and just kind of dabbling in. Same with NKs, I'm just kind of dabbling in, just just now getting my feet wet.
Yeah, well, I think there's a big area there to fill. Like you said, I don't know much about it. I don't know that there's much known about the how regulation of chromatin influences short versus long-lived lineage commitment. So I kind of there might be something there to look at.
Absolutely. Yeah, I'd definitely be interested in looking into that for sure.
Okay. Thank you.
No, thank you.
I got one more question. So if you were to take, okay, so I know you're activating the cells. If can you take away the stimulus and have them recondense their chromatin?
Oh yeah, that's a great question. The only way that we know how to stop condensation once it's in process is by chelating calcium. That seems to be, and in fact, given how fast it works, and so we have been able to see decondensation happen within 15 minutes, as I showed you earlier. Suggests that one possibility is that calcium could have signaling independent roles. In other words, they could be acting on other molecules like structural maintenance of chromosome proteins, or even acting on the chromatin itself. I mean, it is a divalent cation. And so people have shown just with beads on a string DNA, you throw calcium on it, it will spontaneously condense. So calcium could be acting directly onto the chromatin itself. We're definitely interested for sure.
I have one more question about I dig the alpha beta gamma delta comparisons you're making, and it gets me kind of wondering about the specificity of antigen and how that TCR its antigen specificity influences condensation that's going on. So I mean, very simply, I think of alpha beta being very 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 diverse TCR repertoires, and gamma deltas being much more restricted. So I mean, in the context of alpha beta, it makes sense to me. The better binder is going to get more activation signal and deconjugation stronger. But in terms of gamma deltas, what do you think is different?
No, I totally agree. And and for the sake of our experiments, we're trying to eliminate as many confounding variables as possible. We're taking our gamma deltas from the blood so that we're getting them from the same environment. Who's to say what would happen if we were to go into the gut and get gamma deltas? We could get an entirely different story, because you're absolutely right about gamma delta. So for those that don't know, different clones of gamma delta T cells will occupy different tissues. So you can actually, you can almost take a heterogeneous population of gamma deltas. If you have the right surface markers, you can predict where that gamma delta came from just based on the surface markers that are present. And I wouldn't be surprised at all if they have different kinetics. Now, that being said, the way we're activating our gamma deltas is by combining PMA and ionomyosin. So we're not actually stimulating the gamma delta receptor directly. So we're stimulating those downstream molecules.
Thank you.
These have been great questions. You guys are awesome.
Well, I thank you for being our first guinea pig, Jason, and being willing to give up your Thursday night to come out here and talk to us. And I'm hoping that as we go, people will get interested, and people will tune in, and this can turn into like a great way for us to get together outside of just those, a couple meetings that we have here and there. Oops, I hit the wrong one. Oh no, I wanted to share my screen because I had there it is. No, there it is. Before people take off, we can certainly hang out and have a little more chit chat, but before people get out of here, I wanted to see if you all will mind giving us a little bit of feedback. Since this is the first time we've tried this, we'd like to know what you like, maybe didn't like, or if you have suggestions about things that we could do differently. We would love to hear that. And then that way we can continue to improve our Thirsty Thursdays as we go forward.